Hello everyone. Hi. Hope I'm audible. Can someone confirm it? Yes, can you hear me?
Yes, ma'am.
Yeah, thank you so much. So, good evening everyone. It's really great to meet you all this evening. My name is Ponmati and I'll be taking a part of your Omics internship program. Omics is basically a highly diverse subject. Since my PhD was on cancer genetics, I will be sharing most of the concepts that are related to cancer genetics. So to begin with, on behalf of NyBerMan team, I welcome you all for today's session, and we encourage all the students to be more interactive, which means whenever you get any query or any doubt, you can always stop us and you can question us, okay? So before I start, can I have two quick answers for why or what was your interest in joining this session? Very quickly. Let's not take much time. If you feel, if you don't want to unmute yourself, you can just put it in the chat also. So what was your basic idea or interest in joining this?
To
Anybody?
have a better understanding of next generation sequencing and analysis of sequences.
Great. Okay. So you basically wanted to have some idea about NGS analysis. Great. Good. Anybody else would like to add to it? Okay. Okay, so everybody please do feel free, okay? Yeah, so in this session, I will be more emphasizing on the importance of differential gene expression analysis, okay? So can somebody tell me or can somebody just brief us on what is your idea about differential gene expression? Very fastly, again. Anybody? Do you have any idea on what is differential gene expression analysis? Yes, I have some answers coming up. Yes, Abdul. Just go ahead. What is it? Okay. Okay, so probably this is your first class. I really don't want to keep asking much questions. Let's just go into the very basic. Okay. Yeah, that's okay. That's fine. Absolutely fine. So, and I would like to go from the basics. So I guess that should be very much fine. Yeah. So, well, so to begin with, differential gene expression analysis is something that deals with identification and analyzing the gene expression within the context of its difference, which I will be explaining in much detail in the upcoming sessions as well as in the upcoming slides too. Okay, so I just would like to give yeah, exactly, Abdul. Yeah, you are very good. You are absolutely correct. Exactly, Joy. Exactly. Beautiful. So, very good, very great. Yes. So, it is all about studying genes expression. Sometimes it is all overexpressed, sometimes it is all down-regulated. Exactly. Cool. Exactly. Nice understanding. So, let's just go with it. And to just to cover all the audience and to make everybody to understand it more clear, I would like to give you a very brief introduction about what is gene expression. Okay, so what is gene expression? Yeah, so we have been looking into this particular picture for a very long time, which beautifully explains us this concept called central dogma, right? So what is central dogma? So central dogma is nothing but wherein the nuclear material which contains the DNA goes through this process called transcription, giving us mRNA through a process of what is called as transcription. And upon translation, we end up getting the proteins, right? So why this DNA has to be first converted into RNA through a process called transcription. And yeah, before coming or before giving us proteins, what could be the reason? Yeah, so DNA, exactly. Great. Okay, so I'm getting some answers then and there. That's really great. And yes, so the major reason is, as we all know, the DNA contains mostly the junk part or the non-required part. Okay, so we call this basically as introns. So which is well shown here as green in color, followed by blue regions, which actually code for something or mean something, which we call it as exons. So when your gene gets expressed as protein, you do not want this unwanted region called intron, and you want only just your exons. So the process or the way through which you end up getting a matured RNA and the messenger RNA without any introns is called as splicing, and before which your DNA is transcribed into RNA. So this is your central dogma. And now I hope you people understand what is splicing. So now I have a question. Okay. So let us say I need more expression of a protein. Let us hypothetically put it this way. So let us consider that I need more expression of a certain protein called keratin. So this particular protein called keratin is much involved in certain things like hair follicles, nail regions, etc. Okay, so now I need more amount of this keratin protein. Now my question is, if this protein has to be expressed in abundance, then what should happen to my mRNA copies? Any idea? What happens to your mRNA copy level or copy numbers?
It should be more.
Exactly. Cool. Okay. Exactly. So when I want a particular protein to be expressed in abundance, obviously the mRNA copies corresponding to that particular protein should be more. Great. So you call this particular phenomenon as upregulation. Okay. The vice versa will be called as downregulation. So I just wanted to make this particular gene expression concept clear over here. Now, moving on further, here I would like to say that within your own system, the gene expression is going to be different depending upon the context. Okay, so even so, considering within your own system and the gene expression is going to be really that different, imagine how far the difference is going to be between two different individuals. Okay, yeah, then it means that the difference is going to be really high, right? Now, the same thing goes for diseased condition. So the point over here is there exists some kind of difference in gene expression between a normal individual and the diseased individual. So that is a point over here. And the gene expression we expect to be very good or correct in terms of, is there any interruption going here? Pradanya? Okay. Okay, if you have any questions, you can ask me. Okay, so if I just don't want to break the flow anymore, and I guess we can have some questions then and there, and I'll just go with the flow. And if there is really something very important, please do stop me. Okay, yeah. So now I was telling or talking about the gene expression difference between a normal individual and a diseased individual. So in a normal individual, we expect the gene expression to be very perfect and correct, whereas it goes somewhere wrong in case of our diseased condition. So this study of difference between gene expression between two different conditions is basically called as differential gene expression. Okay, so hope you understand now what is differential gene expression. So why do we need to know, or why do we need to study this differential gene expression? Since it explains us the difference between gene expression, difference in gene expression between two different individuals. This helps in solving many important questions. So what are these questions? Question number one, what is the difference between two different individuals in the context of gene expression? We need to know it, right? Number two, how do I target this difference, right? So the basic answer to all these questions is going to be differential gene expression analysis. And only if we get answers to these questions, it would be possible for us to find a solution or find a therapeutic answer to any disease. Since I am a cancer person, I generally focus on cancer and related questions, but it actually goes well and goes good with any disease. Okay, so understanding or getting answers is possible through your differential gene expression, and hence we do differential gene expression. Okay, so the basic objective is to address these questions through multiple tools. Approaches that give us information about the are nothing but arrays and sequencing. So these are two platforms which provides us information about gene expression. So what is an array? Am I audible? Is it happening to everyone or just to any
No,
other?
ma'am, you are audible.
Yeah, thank you. Thanks for the confirmation. Yeah, thank you. So then let's just keep going. So as I was telling you people, these arrays and sequencing are completely two different platforms. So what happens in array is, as I was telling you people about the mRNA levels, you basically quantify the gene expression in terms of your mRNA copies using fluorescent probes, which are basically attached to a solid surface. So this comes under this category called arrays. And in case of your sequencing platform, that is entirely different. So what makes it different is you basically open the whole genome and you try to read it out from A to Z, which means you are deciphering all the nucleotide sequences and you are trying to understand what went wrong. Okay, so both these platforms gives us some information about differential gene expression analysis, and they will help in understanding the disease better. So what is the meaning, or why do we need to understand the disease better? So when we understand the disease better, we try to find the therapeutic options fastly, and so that the survival is increased or a better survival is provided to the individual who is suffering from a disease. Okay, so this is what is array, and this is what is the gene expression tools array and sequencing does to us. But our program will be more focused on how to retrieve some data from these platforms and how to analyze them to make some meaning out of it. Okay, now moving on, I'm going to now talk about some repositories which basically give us some information about all the informations that are generated. Okay, I'm getting some questions. Okay, ma'am, will you repeat about array again? So probably at the end we do it. I don't want to break the flow much. Array use probes that tells us about the gene expression. Exactly, Abdul. Great understanding. Good. Great going. Okay. So now, what are databases or repositories? So as we all know, NCBio is a beautiful repository that we biologists keep using. So it is a repository for what? Many informations, right? So these repositories or databases store the information about patients, which can be retrieved by us and we can explore them further. So now the question is, what kind of informations are stored inside these repositories? So here we have a variety of informations which are stored in the repositories. So let's say, for example, when I am going to explore a cancer related repository, these are the list of informations that I may get. Number one, DNA mutation. So what is a mutation? So I hope everybody knows that each triplet is considered as a codon, and each codon codes for an amino acid. So any mutation in this triplet codon causes a frame shift. So what happens when there is a frame shift? When there is a frame shift, there is no more normal protein getting translated. there we end up getting abnormal proteins or dysregulated. So that gives us a, I guess somebody's mic is switched on. Can you just check it out? And yep, thank you. So when there is a mutation, there is a frame shift which causes or which results in basically translation of abnormal proteins. So what is so special about this abnormal proteins? When there is a normal protein, normal phenotype, no disease. When there is a abnormal protein coming up, no more normal phenotype, we end up getting a abnormal phenotype or a diseased phenotype. So information about all these mutations followed by copy number variation. So what is copy number variation? So in the beginning, we were talking about the mRNA levels, right? So higher, lower. So when you have, when we talk about copy number variation, we basically are concerned about two different things. One is amplification and one is deletion. When there is higher copy number for a certain region, it corresponds to or it correlates with the disease. We call this as amplification and vice versa for deletion. Okay, copy number amplification and deletion. Then followed by gene expression. I guess we have discussed this, discussed about it extensively, right? Upregulation and downregulation. So when there is a change in this gene expression between two different individuals, especially between a normal and a diseased individual, we are really worried about it, and we would like to explore it more. Then DNA methylation. So DNA methylation is something that is associated with histone modifications, wherein methyl groups are added. So this again alters your gene expression. So far we have been concerned about only the DNA, RNA, protein, but even the microRNAs play crucial role in regulating the gene expression or altering the gene expression. So now that we get all these information about a patient who has gone through any specific disease inside these repositories or genomic projects, we not just about their genomic aberrations, we also get something very important called as clinical data. What is this clinical data? So let's say, for example, a genomic aberration of a specific tumor patient, if it is submitted in a repository, you also get the clinical data of that specific individual, by which I mean you end up getting the details of the sex of the individual, whether he is a male or female, followed by stage of the cancer. Okay, since I'm talking about the cancer condition, so the stage of the cancer the individual going through matters a lot. So you get the information about the stage of the cancer, followed by the therapy. Let's say, for example, in which particular round of chemotherapy the specific individual was on during this specific gene expression or retrieval or exploration. So all these data and all these informations are available for multiple samples and multiple individuals, which are stored in many databases or repositories, which in course of time we will be learning to explore and make some meaning out of it. Okay, so that is our end point or objective. Okay, now moving on further. So as I told you people just now, we basically collect samples from patients, right? And then we do their gene expression and we end up getting multiple information from them. So those individuals who basically gave us samples to understand the disease better, we call them as patient cohort. Okay. So what are the things that we learn from them? So once genomic expression data has been created, we end up getting huge amount of data. So we know the expression pattern of the individual. We know what are the copy number alterations that is going in that specific individual. We know what are the somatic mutations that are going on, along with epigenomic profiles, by which just now we have seen what are epigenomic profiles. It corresponds to DNA methylation and other things. And then finally, structural aberrations. Structural aberrations are nothing but the abnormal proteins that are generated. So these many informations are created, or we have been given with these much amount of data at the end of the cancer genomic projects, not just cancer genomic projects. Again, I'm stressing for any disease it is available, but I generally focus on cancer. Okay. Now, so just here you have some of the cancer genomic projects which are very much active and which give you such information that I have been telling you so far. The very great ones are the Cancer Genome Atlas and the International Cancer Genome Consortium. Then you even have Array Express and Gene Expression Omnibus, followed by Catalog of Somatic Mutations, Cosmic. Okay. In course of time, we will be going through some of them. Okay, few of them. So when I talk about this International Cancer Genome Consortium and the Cancer Genome Atlas, the major objective of this specific genomic project is they have obtained full catalog of the genetic alterations in 500 tumors from 50 tumor types. Can we just imagine? So there are, as I can just go back and show you how, as far as the location is concerned, we have many types of cancer going on. So the type of cancer that goes in your brain, you call it as glioblastoma. The types of cancers which affects your bone marrow, you call it as leukemia. The one which affects your stomach, you call it as gastric cancer. So all I just would like to tell you is there are multiple cancer types which exist. So cataloging the genetic alterations, not just mutations. Imagine they are covering the whole genetic alterations, including copy number alterations, epigenomic profiling, etc. and etc. So all these are being cataloged and it is all really readily available in these consortiums and repositories. Okay. So this is actually a very basic slide to explain you people. I guess most of you should be very much good with this, but if there are any beginners, I would like you people to understand these very basic concepts. So I would just really brush it up. But if you are still more interested, you can really find good references which explains all this. Okay, so since cancer is a disease which is acquired majorly through mutations, spontaneous mutations. Okay, throughout. The percentage of cancer being acquired through genetics is really less. Only your lifestyle patterns and the somatic mutations which you acquire during your routine course of life basically leads to cancer. Okay, so it is really important to understand what are the basic genetic alterations that are going on. So when I talk about mutations, so when there is a single nucleotide change, I call it as, or we call it as point mutation. So this single point mutation can be either an insertion or deletion. So when there is just an addition of a nucleotide, that is called as insertion. And when there is a removal of one nucleotide, you call it as deletion. So the point over here is, as we have seen just now, this kind of insertions and deletions basically alters the frame of your whole coding region. Okay, so we call this as frame shifts, which results in producing abnormal proteins. So these are very critical and crucial in cancer. Or the process of cancer formation, we call it as carcinogenesis. Okay, so it is going to be really very dangerous, the mutations. Okay, then you have deletions, as it is very much evident. A loss of a particular segment can really have a very lethal effect on your whole cell cycle and the division process, etc. So deletions and then extra copies, what we call it as gene amplification. Okay, it basically corresponds to increased cell cycle, or it basically corresponds to resistance to a chemotherapeutic agent. So amplifications are again dangerous and followed by structural variation. So I'll be explaining about the structural variation in detail in the upcoming slide. But here to just give you an overview, you can see very well that a chromosomal segment is being fused. Okay, so it is just fused between between two different chromosomes. This basically alters or it creates new proteins, and that actually drives your cancer process. Okay, and then you have something called as mutations in your epigenome. So as we all know, your histones are proteins which are wrapped around your nucleic acid or DNA. So when there is any mutation in the gene that codes for your histone protein, that will affect your whole cell growth and division process, because if histones are coated over your DNA is not open for your gene expression. If there is no histone in a specific region, then your DNA is actually available for the transcription process. So the point, or in a very short, all these alterations contribute or drive cancer. Okay, so I have just given you superficially. If you want to learn in detail, you have really good reference books for it. Okay, so now moving on further, why do we need to study cancer genomics? Okay, so we have this question. So what would I end up getting or studying cancer genomics? So as I have stated earlier, understanding such complex and dangerous diseases is really important, because only if you understand the molecular mechanism that drives these diseases, you will try to find a new therapeutic option for them. And not just therapeutic options, even earlier diagnosis becomes very important in such critical diseases like cancer. So it is really very important to understand and study cancer genomics. So primarily, as stated over here, you need to find a new cancer gene which is driving the cancer, right? So once I have a gene which drives the cancer, I know how to target it. Or now that I have identified a therapeutic target, I can think of the way to overcome this therapeutic target, and then identify molecular signatures to stratify tumors. So I guess I can take a minute to explain here what is molecular signatures basically. Okay, so let us say there are, so now I'm going to enter into a hypothetical example so that everybody understands what is a molecular signature clear. Okay, so just keep following me. So let us consider that you have 25 genes. Okay, so when I compare the expression level of these 25 genes between a normal individual and a cancer individual, I could see that 13 of these genes are abnormal in cancer individuals. Okay, let's say hypothetically that these 13 genes are upregulated. So whenever a patient comes for diagnosis, and when I have some gene expression anomaly in these 13 specific genes, I can call them as positive for my diagnosis, and we can plan for a treatment option earlier. So it is just a very layman and very simple example, but molecular signature actually means more than this. Okay, so when we create or when we generate such molecular signatures, it really helps us to classify tumors further. Okay, so when I classify my tumors, let's say for example, I know 5 individuals have a specific type of tumor. I'm going to further classify them like 3 as tumor type A and 2 as tumor type B, which show different molecular phenotype. Then it is easy for me to make medicines or make therapeutic options differently, so that these two groups which differ at molecular level respond to my therapeutic option so positively. So that is the idea of these molecular signatures. I am pretty sure, don't worry about it. We'll be learning more about this and understanding it more clearly in our upcoming sessions also. Okay, so if you have got what it is, then well and good, great, keep going. Okay, so now, yes, I have identified all these new cancer gene, and I have found a new therapeutic target, and I now know what is a molecular signature to subclassify my tumor type. What next? Very simple. Now I have a personalized cancer treatment ready in my hand, using which I can treat individual patients in a unique way so that they respond to all these treatments so positively. Okay, so that is the idea of this kind of cancer genomic projects and cancer genomic questions that we have in mind. So at the end of the day, we want some successful therapeutic options, or at least a diagnostic assay or a tool that would help us for earlier diagnosis, right? So now, yes, I would like to explain you one of the well-known genomic alteration that drives this specific type of cancer called chronic myelogenous leukemia. Okay, so I hope many of you would have heard about it and even studied about it, but let's just look into it, what it is. So this, what is it? CML, that is leukemia, is basically caused due to a reciprocal translocation between these two chromosomes, 9 and chromosome 22. In a normal individual like us, we have a normal chromosome 9 with ABL, which is very far away from chromosome 22. But what happens during this reciprocal translocation is this ABL from chromosome 9, it goes and sits with BCR in chromosome 22. This is not good. This is basically very bad. This kind of reciprocal translocation drives the cancer. So let's see how this happens, how this mere translocation of two segments led to leukemia. See, when there is a translocation, so I'm just going back for a minute. See, when there is a translocation that is happening, which means, so these are chromosomal segments, and I hope everybody understands that these are just nucleotide sequences. So during the process of transcription, they end up creating messenger RNAs. So as you all see, as you all can see over here, you end up having a fusion mRNA which contains both these regions, BCR and ABL. So what happens or what leads to this kind of a leukemic condition is this BCR ABL messenger RNA has a domain called SH1, which actually creates a constitutive active receptor for the enzyme tyrosine kinase. So I really don't want to go into much of the technical aspects. So for those who keep reading about the cancer biology, should be understanding what is tyrosine kinase. So when you have a constitutively active tyrosine kinase, it actually drives your cell cycle in a very fast mode, so that we end up getting tumor. Okay, so to make it very simple, to put it in a very simple term, let me explain that this kind of fusion basically it creates a domain which makes the cell cycle progression so active. Now that we have identified the causative factor, it becomes very easy to target it, right? So for the first time, the targeted therapy that has been established is this imatinib. It has been really a very great boon, because this kind of leukemia was actually affecting more children. So children actually required lots of successful therapeutic interventions, and this was actually a very great boon. So now that the genomic aberrations are identified, you end up having a therapeutic target for it, okay, therapeutic options for it. So we call this basically as targeted therapy. Okay, so now to move on very quickly into a very another example, we have something called as BRAF gene. So I guess most of you people would have been looking at this BRAF gene, and this majorly is found to be mutated in a specific type of cancer called as melanoma. So this is basically when it gets out of control, it causes increased cancer cell growth. So you end up getting a targeted therapeutic agent which actually intervenes your BRAF gene, so that your BRAF is kept within control and there is no more increased cancer growth cell. Okay, so these are some examples of targeted therapeutic options which are available in today's routine. Okay, so we have all these in clinics going on. Okay, so now very quickly to summarize, what is targeted therapy? Now in this picture, look into all the individuals. So you are basically looking at individuals who have same type of cancer, but their gene expressions are different. So when I plan to treat them with same type of therapeutic option, that is not a good suggestion, or that is not a good option, right? So target 1 is found in certain individuals, target 2 is found in certain other individuals, and no target has been identified in some other individuals. So the best or wise idea could be targeting the options, or that using the therapeutic options which will target your 1 and 2. should be the best options. So this is what we call it as targeted therapy. So even within, okay, so let me just, for those who are beginners or who are very new to cancer biology, I would like to tell you people that, so it is not the way that we imagine. So if, let's say for example, patient A goes into a clinic, he is not given the same medicine or same chemotherapeutic option as all the other individuals. Okay, scenes are changing nowadays. So patient A is different from patient B, and hence they are actually treated in different modes. Okay, so how they are treated in different modes? Basically, we try to understand what is a target that is different among these individuals, and then we take that target and try to identify the appropriate therapeutic option for that target. So this is what is targeted therapy in a nutshell. And for those who do not have any targets, no other option, they end up getting a conventional treatment, which involves either surgery, chemotherapy, or radiation therapy. Okay, but with this genomic era, I'm pretty sure most of the individuals whose genome will be very pretty much sequenced, and they will end up getting targeted therapy, and all these conventional treatments would have been preceded by the targeted therapy very soon. Okay, so now in our internship program, we are going to majorly focus on cancer bioinformatics. So why cancer bioinformatics? Data generated through sequencing and microarray are so huge, which are put into all the repositories. Now it is our job to analyze them and to make some meaning out of it. As I repeat it again and again, yes, we need some meaning out of all these huge data. That is our idea. So in biology, what do? We frame some hypotheses, we ask some questions, we collect some samples, and we perform some experiments. So in the case of informatics is concerned with storage and retrieval of data, and to visualize some beautiful data using some tools and using some computational methods to analyze some important statistical concepts, and finally to validate your data using appropriate statistical methods. So in cancer bioinformatics, we try to compile both these domains to make some information which would help in diagnostics as well as in treatment. Okay, so moving on. So when a specific individual is being sequenced, so as I told you people, what is the meaning of sequencing is nothing but you literally decipher all the nucleotide sequences present within the individual. So you compare it with a normal individual and we look for somatic mutations. Okay, this is done. Yes, there are mutations here and there. How do I identify which specific mutation was contributing to my carcinogenesis process? So that's a very big question, you know. So in order to answer such questions, we need to have some tools. So that makes it very interesting and important to study cancer genomics. And then I guess in the previous slides, I used this word called stratification. So let me explain it now. So what the picture, whatever you people are looking at in green and red color is nothing but a heat map. Okay, so it is nothing very complicated. It is very simple. Heat map is basically a visualization. Okay, so the way you look at it. What do we understand from this heat map? Yes, here the patients, or okay, so since we are concerned about the cancer patients, and since we are discussing continuously about the cancer informatics, so let us just keep again the same example of cancer expression over here. So if you people remember, we had expression pattern acquired from multiple patient cohorts. Now, is there any expression overlap between the individuals? Are they showing any correlation in terms of their gene expression? That is very much understood with the help of heat map, which is basically generated through hierarchical clustering. And I guess in one of the module, we have this hierarchical clustering and how to visualize or how to make such beautiful heat maps using the gene expression data will be taught. Okay, so understanding and doing heat map is a different thing. In this particular theoretical session, I would like to explain you people the importance of heat map. So what do we understand from this heat map? Very simple. Patients showing same type of expression or grouped as a subtype, or that is what is the word stratify mean. Okay, you basically classify your patients based upon the gene expression into multiple groups. Very simple. Once if I have understood the gene expression, and then if I have grouped my patients based upon their gene expression, now I know what or the therapeutic options that could be looked for them, and then it becomes very easy to treat my patients as a group, okay, as a subtype. Okay, so let's say let me just give you a very small example over here. So here you have breast cancer. So when somebody tells when somebody has been diagnosed with breast cancer, so breast cancer is a type which has multiple subtypes. Okay, so the multiple subtypes include estrogen receptor positive, PR positive, progesterone receptor positive, Herceptin, many what is it, hormonal receptors make subclassifications, and beyond that you have many other molecular subtypes wherein the breast cancer patients can be grouped based upon their molecular pattern or the molecular expression pattern. So based on it, we have luminal A type, B type, and many other subclassifications of breast cancer. So this kind of stratification for breast cancer was possible just because of this hierarchical clustering, which was done on the gene expression data obtained through microarray analysis. So when we study or when we get to know the molecular expression pattern of these many breast cancer patients, we can very easily try to identify their progression level or how do they do with some kind of therapeutic options and etc. So these are the very good applications of your cancer informatics. Okay, and some other applications include, I guess we have discussed it very much enough, right? So once you know what are the genes that are getting modulated differentially in a diseased condition, you can end up doing a pathway mapping, you can go for target identification, you can go for gene screening, disease characterization, understanding the disease much better, and finally we end up getting good, successful, personalized treatment options. Okay, so that is the end point. Okay, so this again is a heat map which actually explains or it actually explains how the breast cancer patients are classified based upon their molecular expression pattern. See, normal like luminal A, luminal B, HER2 basal. So these individuals, they differ. So for those who are looking at heat map for the first time, your column represents each individual, and each row over here represents a gene. Okay, so in the upcoming classes, I will be explaining more about how an expression file looks and what is the format of your expression file, what is the frame of your expression file. Everything I'll be explaining. I'll be explaining in much detail. Okay, so just keep traveling with us, and we'll just try to explore more. Okay, and yeah, so these are the very significant applications of studying gene expression or cancer bioinformatics. And now moving on. Yes, I guess we are coming to the end or the conclusion part. So I guess so. This is going to be the plan, or this is going to be the modules that we have planned for your internship program. So the beginner level covers something that is related to RNA sequencing, a step-by-step guide to RNA-seq, a computational analysis method, followed by introduction to small RNA sequencing computational analysis. Okay, in 30 days module, you will find more of the cancer informatics, where I'll be taking more in detail classes on how to extract microarray data set from gene expression omnibus, and then how to do a differential gene expression analysis, and then how to make a functional meaning out of it using the tools whatever I have mentioned over here, followed by, as I have told you people, we will be doing some hierarchical clustering using using tools like dchip, okay? And also I will be exploring, or I'll be taking you people to learn more about the consortiums or the repositories that I have discussed so far. And then, yes, in the 45 days level, we call this as where there's a mix of both. That actually we haven't included, I guess, but yeah, there will be a mixture of more of RNA sequencing analysis along with the microarray analysis tools that we have discussed. Okay, so this is what is your modules are going to be. I hope you people find it really informative in the upcoming sessions too. And just before we close, I just have a very small, I don't know how much you people like it. Yeah, we have a very small quiz time. I just want to know if you people have been traveling with me so far. Good. Where, sorry, we can find. Sorry, I just take this question. Yes, yeah, yes. Yeah. So I think the organizers will be letting you know, you know about the recordings of the session very soon. Yeah. So just to know about the today's session's understanding level, I would like to ask just few questions. Okay. So can I have a volunteer to read out the first question? Anybody? Would you, no need to give the answers, just you can read the question along with the options.
Yes.
Yes.
I guess, I don't know.
Yes.
Yes. Let the first question be done by Abdul, followed by the second question by Ananya. Would that be fine?
Yes, it will be fine.
Great. Go ahead.
What is transcribed form of DNA to produce a protein, and it's good indication of whether or not a particular gene is expressed?
Yeah, I started getting answers very fastly. Yeah, you just go ahead with the options. Yes.
Option 1, mRNA. Option 2, cDNA. Option 3, tRNA. And option 4, tRNA.
Tivuk, this is really so wrong, you know. Before starting the next question, even then you have started giving the answers. Okay, so I take that as so much of interest, and I take it so positively. Yeah, thank you so much for all the answers, and kudos to everyone who gave the right answers. Yes, mRNA is the correct answers. Since everybody gave the correct answers, and since there was answer even to the next question, I'm going to ask you people one more question, okay? So which is not over here. So listen to me carefully and give me the answers to that question too. Okay. So now there are two individuals, A and B. Okay. This is again a very simple question. So individual A is actually having COVID infection. Individual B is healthy. Now both of them are screened for COVID virus. So what will be the level of mRNA that corresponds to COVID virus in individual A? How much? I don't know if I went very fast. Let me repeat it slowly. You have two individuals, A, B. A with COVID infection and B, healthy individual. Both of them were taken swab test to confirm their infection. Okay. So now the question is, what will be the level of mRNA that corresponds to coronavirus in individual A in comparison to individual B? Yeah, exactly. Sachin, Priyanka. Exactly.
Overexpression.
Exactly, Soumya. Thanks to everybody who were actually following with us today, and thank you so much. And Ananya, would it be will it be fine if we could go with the next question too? Just go ahead, Ananya. Go with the next question. We'll just finish it off. Ananya.
The main contributing factor for most cases of uncontrolled cell division that occurs in human cancers is 1. Mutation of genes, 2. Replication of genes, 3. Transcription of genes, 4. Translation of genes.
Exactly. So I got answers already coming up. Yeah, exactly, Faisal. Exactly, Amir. Yeah, Kartavya.
Mutation.
Exactly. Thank you, everyone. Exactly, mutation. Yeah. So thank you, everyone. I hope you will like today's session, and I hope you people keep traveling with us, and we'll be learning more and more in future together. Yeah. So with that, I guess we can wind up today's session. And I guess you people can just wait for a minute